So physiologically then, an increase in venous return relates to an increase in the filling of the ventricles, as there's an increase in blood returning to the heart to fill those ventricles. And if the ventricles fill more, the heart has to increase not only the stretch of the heart, but also the force of the contraction to force the blood from there. Furthermore, with a larger force of contraction and an increase in the amount of blood pumped from the heart per beat, comes a larger stroke volume. This is called Starling's law. So Starling's law is an increased stretch of the heart, and that's in correlation with a larger filling of blood in the ventricles of the heart, like we previously mentioned. Therefore, at the start of exercise, there is an increased stretch and volume of blood in the heart due to a higher demand for oxygen at the muscles. So obviously the higher demand for oxygen at the muscles means that the heart has to force more blood out and increase the force of contraction to increase the pressure and volume of blood pushed through the arteries to those muscles to increase the amount of oxygen supplied to the muscles. Much like the other factors, this plateaus after about 3 minutes as the demand for oxygen is met, so there is no need for an increased force of contraction or an increased volume of blood. And finally, after exercise finishes, this should return to normal. The contractions should return to a normal force and the blood should fill to a normal volume, because stroke volume similarly returns to normal and cardiac output returns to normal. So thermoregulation is the last factor of the cardiovascular system that I will discuss. The production of heat as a waste product in the conversion of food into energy for muscular respiration is one of the factors that will contribute to steady state exercise. So this waste heat that is produced as a product from the conversion of food, this is produced at a higher rate due to the more regular contractions in exercise. So the more regular contractions means that the muscles are having to convert that food at a more regular rate, meaning that heat is produced as a waste product more regularly, which is why that waste heat is produced at a higher rate during steady state exercise, because the muscles are undergoing regular contractions, meaning that waste heat is being produced regularly. So the actual thermoregulatory response to this production is that central thermoreceptors detect this change and send a message to the hypothalamus that sends a message to the skin to respond. The skin responds by flattening its hairs to release heat, as when the hairs are raised, it actually traps the heat, so as they flatten, this allows heat to be released. Blood vessels at the skin also dilate, so there's more blood flow in colder areas of the body to cool down. So obviously, closer to your skin is going to be closer to external temperatures and away from the centre of your body, which is warmer. Therefore, due to the blood flow being in colder areas, your body can cool down. And finally, you also release sweat as a response, which is the release of water and electrolytes on the skin's surface in the form of moisture, which evaporates from the skin's surface. Moving on to the respiratory system then. So if we first look at tidal volume, which is the amount of air inhaled in a normal breath. Tidal volume initially increases at the start of exercise due to an increased demand for oxygen at the working muscle. So obviously if tidal volume is the amount of air inhaled in a normal breath, your body is going to forcefully inhale more in your normal breaths because there's an increased demand for oxygen. So if you inhale more, you're going to increase the inhalation of oxygen, which then obviously travels from your lungs to the heart, through the blood to the muscles. This obviously allows a larger supply for oxygen, which allows those more regular contractions in the increased intensity of exercise. Similarly to the cardiovascular system and those factors, after approximately 3 minutes, tidal volume begins to plateau, as again there isn't an increase in demand for oxygen. Therefore, you don't need to inhale more oxygen. At the end of exercise, tidal volume will decrease as there is a lesser demand for oxygen due to the oxygen being sufficiently supplied. There is no need for that continual heightened supply of oxygen, as any signs of any lactic acid will already be rid of, and carbon dioxide should hopefully be rid of as well. So that return to normal tidal volume should be fairly quick. So breathing rate is really similar to tidal volume in the sense that breathing rate initially increases at the start of exercise due to an increased demand for oxygen at the working muscles. So much like tidal volume, increasing the amount of air inhaled will increase the supply of oxygen. Increasing the regularity of inhaled oxygen is going to increase the supply of oxygen at the working muscles also. After approximately 3 minutes, breathing rate begins to plateau, as there isn't an increase in demand for oxygen, so the breathing rate does not need to increase, and you don't need to increase the amount of oxygen as there is sufficient supply. At the end of exercise, breathing rate will decrease again as there is a lesser demand for oxygen due to oxygen being repaid already very quickly, as the demand for oxygen shouldn't be as high as that of perhaps anaerobic exercise. Now, the average breathing rate is 12 to 20 breaths per minute. Elite athletes will have a lower breathing rate, similar to a lower heart rate. This is again because their respiratory muscles would have been trained, the alveoli would have increased in size through training. Therefore, they don't need to breathe as regularly because they have an increased tidal volume and an increased lung capacity. If we look at the neuromuscular response now then, and this is in relation to Golgi tendons and muscle spindles. So muscle spindles are sensory receptors found in skeletal muscles. They detect changes in muscle length, which then contributes to fine motor control when they relay messages to the central nervous system. They also regulate the contractions of muscles. This happens because the muscle spindle activates motor neurons via the stretch reflex. The muscle spindle detects when a muscle stretches and sends a message to the spinal cord, where secondary sensors detect the degree of stretch. This information is then sent to the central nervous system via the sensory nerve. At the central nervous system, a message is then sent to the motor neuron to cause contraction and resist the stretch. Golgi tendons, at a glance, would seem very similar to this. However, these are proprioceptive sensory receptors, and they're located at the origins and insertions of muscles. And they detect change in muscle tension. So when the Golgi tendon detects a change in tension, it sends a message to the central nervous system, which then sends a message to the muscle spindles. It then interrupts the muscle spindle's contraction and causes the muscle attached to it to relax, meaning there isn't any overstretch or isn't too much tension, which would obviously lead to injury. Furthermore, in steady state exercise, these contractions should be regular without an increase in intensity, meaning the spindles and Golgi tendons are detecting the same changes in tension and length of muscle and are therefore reciprocating with the same response each time. So, perhaps the most important factor for Golgi tendons is actually the start of exercise, because if you haven't warmed up, your muscles haven't undergone those contractions and that stretch and increased tension yet from the contractions of exercise. So the Golgi tendons and muscle spindles work together to prevent overstretch and overtension at the start of exercise when you initially begin those contractions from a resting state, obviously preventing injury. Going back to respiratory responses, the effects of pH, temperature, etc. on the oxygen dissociation curve then. So the oxygen dissociation curve is a graph that shows the percent saturation of hemoglobin at various partial pressures of oxygen. The purpose of this curve is to show the equilibrium of oxyhemoglobin and non-bonded hemoglobin at various partial pressures. At high partial pressures of oxygen, hemoglobin binds to oxygen to form oxyhemoglobin. When the blood is fully saturated, all the red blood cells are in the form of oxyhemoglobin. As the red blood cells travel to tissues deprived of oxygen, the partial pressure of oxygen will decrease. So if we think of tissues deprived of oxygen, initially at the start of exercise, that would be those working muscles as they have insufficient supplies of oxygen. So consequently to this, the oxyhemoglobin releases the oxygen to form hemoglobin. So pH then. A decrease in the pH shifts the curve to the right, while an increase in pH shifts the curve to the left. This occurs as a result of a higher hydrogen concentration. And the rightward shift is actually referred to as the Boer effect. A decrease in carbon dioxide shifts the curve to the left, while an increase in carbon dioxide shifts the curve to the right. And finally, in relation to temperature, an increase in temperature shifts the curve to the right, whilst a decrease in temperature shifts the curve to the left. Increasing the temperature denatures the bond between oxygen and hemoglobin, which increases the amount of oxygen and hemoglobin and decreases the concentration of oxyhemoglobin. Temperature doesn't have a dramatic effect, but the effects are noticeable in cases of hypothermia and hyperthermia. Finally then, the energy system for steady state exercise. So steady state exercise would primarily use aerobic glycolysis. This is because exercise lasts for more than 3 minutes, so the anaerobic glycolysis cannot be used. Your body is working with the presence of oxygen due to the lesser demand for oxygen as a result of the intensity of the exercise. So how this works is that ATP will undergo a chemical reaction where the chemical bonds will break and it will become ADP, and we use that in physical activity. This process happens when there is no phosphocreatine that we can use, therefore we have to use a different system called anaerobic glycolytic system. Because there is no phosphocreatine, there's an excessive amount of adenosine diphosphate. The enzyme phosphofructokinase detects this, resulting in a response of it breaking down glycogen into glucose. Glucose is broken down into pyruvic acid, where aerobic glycolysis begins. This occurs when coenzyme A detects the presence of oxygen, which leads to acetyl-CoA, making citric acid, which can be used for Krebs cycle, which creates hydrogen for the electron transport chain to eventually create 34 moles.